Good evening, everyone. I am Shravanti. Today I'll be presenting a topic of the paper titled A New View of the Tree of Life by Laura Hung. Now, getting into the topic, what is the Tree of Life? The Tree of Life is basically a big diagram that shows how every living organism, such as bacteria, archaea, plants, animals, and humans, are related. And yes, it is important because it gives us a clear picture of biological diversity and evolution. By understanding where organisms fit on the tree, we learn how life started, how it changed over time, and how different species are connected. So now the question is how far it developed over time. Originally, the tree of life was based on physical appearances like shape or structure. Later, scientists used a single gene called 16S rRNA to build better trees. But today, we use entire genomes and advanced sequencing technologies, which gives us much more accurate and detailed tree. Now that we know what the tree of life represents and why it matters, the next question is what were the older methods missing? The older methods missed uncultivated organisms because most microbes can't be grown in lab and because gene-based surveys often fail to detect deeply divergent or unusual lineages due to primary bias and data filtering. So, to overcome these limitations, researchers shifted to genome-based approaches, that is, directly from the DNA that can detect organisms even if they can't be cultured. Now, now we'll explore in this slide the genome-based technologies that allow the authors to access uncultivated microbial diversities and reconstruct thousands of genomes, like metagenomics, that allows scientists to extract the sequence of DNA directly from the environmental samples without needing them to grow in the lab. This makes it possible to reconstruct genomes from many uncultivated bacteria and archaea. Then coming to the single-cell genomics, which helps us to capture the DNA of individual cells using FACS, that is fluorescent activated cell sorting. This technique gives access to lineages that metagenomics might have missed. And the paper also highlights the genomic resolved analysis, not only identifying organisms but also revealing their metabolic abilities, advantages, disadvantages, and evolutionary placement. Now, how do we build a tree? We build a tree using modern genomic tools, like they use 16 ribosomal proteins instead of a single gene, giving a much more accurate picture of microbial relationships. And they also analyzed over 3,000 genomes, each representing a different genus, which greatly expanded sampling. By combining these datasets, the study uncovered new evolutionary connections between all the organisms that older methods completely missed. Now, from the methods we saw in the previous slide, this updated Tree of Life shows some major insights. Those are: the first is the bacteria dominate the new phylogeny while other appears as a much tighter cluster. Then also the tree uncovers huge hidden diversity from uncultivated groups like sepia, which old methods couldn't detect. And also finally, some deep evolutionary relationships are still unclear, showing that more sampling is needed to fully resolve the tree. And all these patterns point us to one major discovery in the Tree of Life, CPR, the hidden bacterial world we were missing. Now, the CPR represents a massive branch of ultra-small bacteria with reduced genomes and often require other microbes to survive. This and these are the almost completely unknown before genomic sequencing. And why does CPR matters? It is for several reasons. As discussed before, these were undiscovered in older approaches. Finally, CPR provides evolutionary clues such as branch length differences and taxonomic mismatches that shows how genome-based phylogeny can reshape evolutionary thinking. Now, coming to the key takeaways of the study, we saw how modern genomic tools completely reshape our understanding of microbial diversity. Genomics allowed researchers to uncover the enormous hidden diversity within the candidate phyla radiation, which we now known forms of major part of the bacterial tree. Overall, genome-based phylogeny not only expands the tree of life but also reshapes how we understand evolutionary relationships across different organisms. In conclusion, this paper shows how advanced genomic techniques completely transform the tree of life. And thank you so much for listening. I'll be happy to take any questions now.